thank you very much for the perseverance in keeping this workshop going. As Norm said earlier and other speakers the past 3 days, it's just a shame that we can't be skiing, but I'm a bit relieved because I think there's no snow anyway. So we have to do this some other time. And I'm honored to close the meeting and that there's still many people listening to me, and I'd be happy to answer questions later on as well, of course. So I will mention genome dynamics, and I will start with why we think that dynamics are important. Talking about a little bit about approaches that are in fixed cells, and then I will move on to live cell imaging approaches. So we all know that chromatin moves despite all those static models we have and all these representations that are based on fixed cell experiments and on dead cells, crushed cells, isolated DNA, everything. So what you see here is a movie, a real-time movie of 5 frames per second of DNA that is labeled in osteosarcoma UTOS cells. And you can see that that's clearly not static. And even over the short time frame, so the movie is running around and turning around every 30 seconds, there's quite some significant motion, even if you don't invoke large-scale micrometer displacements. So why are we interested in talking about this? Can you see my mouse?
Yes.
Okay. It's because so the organization in this nucleus is heterogeneous, as you've heard from other speakers. They're dense and less dense regions. And then when you look at the motion of these dense or less dense regions already a couple of years ago, it was shown that it's unequal and that different areas move within different domains here that have different orientations, different diffusion properties, and different speed. And what we believe is that this information based on measuring dynamics at short timescales can actually inform on what is happening at smaller scales, here loops or protein displacements on the DNA and during transcription or repair or replication. So that is the idea, is that we can have a better idea of what the mechanism is doing to the DNA when we look at real-time imaging. And then, of course, at some point, maybe we can also find out whether if this movement is in any way helping the regulation of these processes, which is something that we still don't know. So the model we use are human breast cancer cells. And we use these because they are a nice model of transcription activation rapidly under physiological conditions. So for the non-experimentalists here, we are not using any kind of artificial viral activator in human cells or CRISPR-Cas9 or whatever you do. This is completely natural. What you do is you starve cells from hormone and they sit there, they synchronize, and they just wait for something else to happen. And when you add hormone to it, hormone are very small molecules and they can diffuse through the cell very rapidly. And when they bound to transcription factors, they're called nuclear receptors because they receive the hormone within the nucleus. These transcription factors called nuclear receptors can then find their target sequences, recruit whatever cofactors they need, and eventually polymerase II to activate transcription. So this is a very schematic view, but it's generally accepted that these nuclear receptors do have a genomic function that requires hormone binding and a conformational change to recruit these factors, whether it's on promoters or distant enhancers. And then they drive transcription. And this involves long-range contacts and remodeling of chromatin. This is what I just said. So these cells are called MCF7 cells for the hormone receptor, estrogen receptor positive cell line. They can be cultured in a plate. You need 3 days to hormone starve them, arrest them. And then you add estradiol as this hormone, very rapidly you activate transcription. And when you wait sufficiently sufficient amount of time, you can do all these techniques that other people and that we currently use to analyze what is happening to transcription. So today I will focus first on a region on chromosome 11, which is the progesterone receptor gene. It's a gene that is responsive to estradiol, and it is here. It's a 90 kb gene. It's transcribed in that direction, so to the left here on the slide. And then you have an adjacent region where there is no open reading frame. But when you look at the entire region here, it's about 500 megabases. You do have a number of these estrogen receptor binding sites that were identified by ChIP and ChIP-seq. And it's quite significant binding to these sites. And these sites do not necessarily correspond to the promoter here, which you can identify using ChIP data of various histone modifications, but they are rather located at what we could call enhancers. So we are interested in this region. Also, when you look at existing data and our own data, you can see that there are quite some long-range interactions that seem to happen. You can do estrogen receptor TRPET or POL2 TRPET, and you see that there's quite some contacts that are happening between this sort of region here upstream of this progesterone receptor gene. So promoter capture high-C is something that we did more recently, and also is able to see some changes in these contacts, but the resolution is very, very difficult to obtain at level with this type of approach. So since we do imaging, a first approach we used is high-resolution 3D FISH. We used phosphate probes, which are shorter than Bach probes, about 10 to 20 kb here. We selected several places, and here I'm showing you an example of 3 phosphates that we used across this area, phosphate 1, 3, and 5. You have example images shown here. It's one plane, a single plane out of high-resolution 3D stacks, and you see that these 3 probes here are, of course, close in space because we have short genomic distances, but they become even closer when you add estradiol to the region in a condition where you think that the gene is transcriptionally active. So the probes are distributed and they become closer. You have a rearrangement when you add estradiol to these cells that were previously unaffected. I must say, these cells are arrested in the G1 cell cycle, and adding estradiol does not allow them to enter into S phase, into replicating phenomena, which will take 8 to 16 hours. So within the time frame, these cells are still in G1. There's no replication going on. We then adapted an algorithm that we published a number of years ago, developed in yeast, to these 3D FISH images, which is called 3 loci. And it's based on mathematical inverse modeling, which allows you to define survival zones between the 3 loci in the same cell. So the advantage of having 3 loci is you can measure the relative distribution independently of the nuclear size and define these survival zones, which give you a radius of displacement. And when you look at this in cells, in the absence of estradiol, you see that these 3 sites here, these 3 phosphate sites within the 500 kb region have quite some freedom to move relative to each other. When you add estradiol at first 45 minutes, and then you look again at 3 hours in these cells, you see that the freedom of movement decreases and they kind of become closer in space. So you clearly have this remodeling of these regions, in particular phosphate 5, which is the one that is on the edge of this regulatory region, coming closer to the body of the gene. And so you have a first step, and then you have a next step. I don't have time to get onto into details, but you do have different places of these enhancers that seem to participate in the remodeling at short or longer time intervals. Then in collaboration with Jörg Decker and Mark Marty Renham, we then set out to look at this using 5C, which is a relatively high resolution approach to look at these remodeling within this area. Again, here you have the map. This is the progesterone receptor gene down here. It's just recapitulated larger so that you can see it easier. This regulatory region with these various enhancers, and that's another gene downstream. We can first quickly see that there's this TAD that englobes the gene itself and this regulatory region, and that is separate from the TAD that encodes the other gene. What Mark Mardi-Renum did using his StatBit algorithm is he built models based on the 5C contact frequency maps. And this is these models that come up here. So you have these sort of wall balls that include many, many, many models that come out from modeling these contact frequencies. From the model, you can see that here is this gene in red down here, and this is the area of the progesterone receptor gene. The models enable you to deduce artificial distances. It depends on the parameters, of course, that you input. But let's stick with one set of models. And here you have the arches that represent contacts that are less than 50 nanometers based on these models here, at least 50% of cells. So you can see that this region here, even in the absence of estradiol, so the gene is not transcribed, has some pre-existing organization. You have specific areas that tend to interact with the body of the gene and not at all with the neighboring TAD, but with areas here within this regulatory region. And that is significant because these MCF7 cells are, of course, primed. So the gene is poised for transcription in these cells. If you add estradiol, it can be activated very quickly. Now, if you compare it to a cell line that does not express the estrogen receptor, so these estrogen receptor negative cell lines, where the gene is silent, this is what you see. So at the left is the same panel you saw before here with its pre-existing structures. If you do the modeling of a 5C contact map in another cell line where the gene is silent, you see that you hardly have any cells that have distances between these regions that are less than 50 nanometers based on the models that Mark built. So if you give a model here, I don't have the image. I should have put it on, but it looks like sausage, really. It's dense, it's a sausage. There's no pre-folding here. So the pre-folding is specific to the gene when it is poised, but not transcribed. So now what happens when you add estradiol? This is shown here in a different type of representation. Here I kind of flipped around the map and I only have half of it. This is what you've seen before. Here is this regulatory region, and you see these numerous contacts here in red that are increased with the gene itself. When you add estradiol to it, this is what you see. First glance, there isn't much happening. But that is what you would expect because the gene is in an area that is already pre-organized, as you've seen before, compared to when it's silent, so it's poised. You add estradiol, so something has to happen very quickly so that within 10 to 30 minutes, you can actually transcribe the gene. So you have to look closer into detail, and then you depict these areas that pop up here and here that correspond to these enhancer sites that then interact either with each other or with the receptor gene itself. So basically we see what we've seen using the phosphate probes along this body here, is that you have some areas that come together. They can be between enhancers or between the gene and these enhancers, and other areas that tend to be remodeled and that even lose contacts here after a while once the transcription process is going on. So when you summarize this in using the models that Mark built, here's what you detect in the bottom panel. You see the loops are still here, but now in 50% of the cells, you see that these 50 nanometer contacts can be increased in red. So you have, again, these sites where you have the polymerase and the estrogen receptor binding that tend to increase the contact, so they shorten the distances. And in blue, you do have some that are decreased here between this side and that side. You clearly have a blue line. So you have some things that come together and others kind of open up. So you have a remodeling of the area that accompanies transcription. And this is what you can see on these models here. On the right side, you have the model in the presence of estradiol after 3 hours. The adjacent TAT is not affected. And then here you have this regulatory region in this pink that kind of folds back up to the purple and the blue. And I'm just going to show you a few images that we did more recently, because these, as you know, these are models that are built on 5C contact frequencies. They are not real. So when you look inside cells, what does it look like? So this is what it looks like. This is Storm using a bug that englobes the gene and the regulatory region. And you can see that you have the gene on one side and the regulatory region on the other. It kind of folds back onto itself. On the left here is in the absence of estradiol, you already have a pre-folded region. And when you add estradiol to it, the distances become here a little bit shorter and you have some kind of remodeling. So, okay, these are blobs, but they look, I mean, not so different from what the numerous models that Mark builds based on 5C structures. And this is what we can detect in cells in 3D Storm. So from to summarize this part here, I'd like to say that we have pre-organized structure that gets reorganized when you add hormone to it. It's probably due to some caging that can be linked to the accumulation of protein clusters. And now we wanted to set out and look at what happens in living cells. So I'm not going to talk about the protein clusters, maybe a bit at the end, but we wanted to know how does it relate to motion. So how can you visualize DNA in living cells? So you want to have a fluorescent tag here. But if you have only one molecule, it's not going to be you're not going to be able to distinguish from all the others that are floating around in the cell. So you have to multimerize it. So historically, we've used the LAC and TET operator sequences for the last 25 years. They have been worth a lot, and we're still using them. They are bound by repressor proteins that are binding very stably, and if they're fused to GFP, they make a little focus. But the problem is when you want to study processes of the DNA, such as repair or transcription, then the polymerases don't quite like these repetitive sequences and the stably bound proteins to it. So this can also create blockages and fragile sites, so breaks and so forth. If you put it next to it, that could be a problem. If it's further away, you don't really care so much, I must say. But if it's near your gene, then you care. And a similar issue actually is when you use CRISPR-Cas9, dead Cas9 here fused to GFP, because you also have to multimerize all these guide RNAs. It's nice because you don't have to edit the genome, but you need to have all this local unwinding here. So we developed a tool that allows us to tag the DNA, inserting a sequence, but it's a short sequence. And instead of multimerizing the sequence will recruit proteins that kind of pile up on it in the safe place. So we call this anchor for anchoring proteins. It can be GFP tagged or whatever other proteins you have. And it's kind of nice that Anna talked before I did, because she already told you about the system, which is actually the bacterial PAR-S PAR-B system. So we call the PAR-S sequence UNC and the PAR-B protein OR, but it is exactly the same system. It is borrowed from bacteria. Our first systems were from Burkholderia, but you can also use other types of bacteria. Sequence is short, a few specific binding sites, which will bind dimers of this PAR-B or OR protein. And once the sites are occupied, the OR proteins kind of bind to each other via protein-protein N-terminal interaction. And then these proteins that bind to their friends, they have a free DNA binding domain, and so they just hook onto neighboring DNA with low affinity. So they pile up in the place for 200 to 400 molecules, creating a fluorescent focus that you can see here in yeast. You can see it in human cells. You can see it in Drosophila cells. You can see it in viruses. And if you want to know more about it, please look at various things that we and others have published using the system. So to go back to our progesterone receptor gene, now we can tag it here right in the promoter. We insert the sequence. We express the OR protein fused to a fluorescent marker. So here this is Santa it fluoresces in red. These PARP proteins will bind to sequence and spread. And so you can see a single focus. So this is a single gene in a human cancer cell genome that has about 75 chromosomes. So you see only one single spot. And you can then track this spot over time and measure dynamics. This is what we did. And this is a first experiment where you can see averages of mean squared deviations over tracks that are 150 time frames over 1 minute. And they are averaged out here. The mean square deviation can see that in the absence of estradiol, we almost see here very free diffusion. When you add estradiol and you measure the dynamics in the same cell, but then we average out 10 cells, you see that the mean square deviation plot kind of drops down. So we were curious about this. It wasn't quite what we expected at the time, but we repeated it. And as you can see, these are averages. I didn't put in the deviation, but there's a clear drop in the mobility here. Problem was at the time is the cells we look at, so these are about 10 cells or dozen cells, you measure the motion of the DNA, but you don't know if the gene is actually transcribed because you add estradiol, you think they're transcribed. Now, if you analyze many, many cells, hundreds for FISH or millions for 5C, they are largely sufficient number of cells that most of them will be transcribed. But if you only look at single cells, you have to have a way to know whether your gene is transcribed or not. Because otherwise, what we see here could just simply be an overall effect on throwing hormone onto a cell and have some kind of a global consequence on the motion. And also alpha is clearly difficult to measure. So then, and this was published a number of years ago, so I will go rather quickly here, is we put it on a transgene, which has only an open reading frame, but it also has the MS2 stem loops, and the MS2 stem loops will enable you to visualize the RNA. So this requires transfecting a marker, the MCP coat protein fused to GFP, which is also fused in the cell here, in addition to the PARP OR protein, which is red and labels the DNA. So here the cells are hormone deprived. The gene visibly is not transcribed because you don't see any green spot. When you add estradiol to it, you expect transcription to occur. And then you have these stem loops of the messenger RNA that can be bound by MCP, which is the case here. You see a green spot accumulate. This is exactly the same cell as this one, only 45 minutes later. So you can measure dynamics here, add hormone, and measure dynamics again. And then you can see what is the difference in the same cell. So these are just sort of some live cell imaging here for you to appreciate how beautiful these look, the DNA and the RNA next to it. A couple of excerpts of frames every 250 milliseconds where you can see that the green messenger RNA actually drifts a bit away. There's no continuous transcription, but so you can track the red spot and you know that it is transcribing. So you have a perfect idea of what you're looking at. So just to really insist that what we do is we look at the same cell, the same DNA fragment, the same spot, here not transcribed and here transcribed. And what we found is that here in blue, you look at the area at which this spot roams here within 200 nanometers, and you add estradiol. We only track the cell if we actually see messenger RNA. If we don't see messenger RNA, the cell is thrown away and you have to start again. And you clearly see that the area is reduced. You can do mean squared deviation on a single cell, a single spot in blue in the absence of estradiol. Again, we have this sort of almost free diffusion. And you add estradiol and puff, you see the mean squared deviation drop down. And this is a case for every single cell we looked at prior to transcription. It has a greater MSD slope than when you add estradiol to these genes. These are an average of 22 cells. You see that you have almost free diffusion for most of the non-transcribed genes, and then it drops down with a much lower exponent. So you affect the type of diffusion that you see, and we have numerous spots here. So on the right-hand side, so what we plotted is a number of cells. Each symbol represents one cell of the MSD value at 40 seconds. And you see that there's huge heterogeneity. Now, when you add estradiol and you transcribe the gene, every time it is reduced. But the original dynamics are really diverse. So one thing I'd like to conclude from this is that the DNA moves quite a lot. When you transcribe it, motion is reduced. In particular, the area in which the gene, the tag locus moves. And the second point is that initial dynamics are quite heterogeneous. So you have an effect of what's happening, but you also have something that will create the initial motion that we didn't know. So this is where Itham and Roman come into play. Itham was a postdoc and Roman was a pre-master student. He's now doing his PhD at Delft in the Netherlands. And Roman, hi, I think you're listening. So I hope I'm not going to say anything stupid here. So what Itham set out is he imaged whole genome motion. You can do this using either H2B GFP or tagged DNA. And then we do this over 100 to 150 frames with 200 millisecond interval. This is just normal spinning disk imaging. It's not high resolution and it's dense molecular labeling. So what they did is they analyzed the motion of each of the intensity of fluorescent of each pixel within these nuclei and applied optical flow to analyze the motion of the variation of the intensity within each pixel compared to the surrounding pixels. So the first study is based on the first series of analyses that are called dense flow reconstruction correlation, showing that they are domains of motion. And what was interesting in this study is that the borders of these domains are very smooth in the absence of transcription. And when you activate transcription, they become rougher. But data you can get out of this quantitative data are averaged out. So the next step was to actually spatially resolve the motion of these nuclei. And I just want to take a minute here and tell you why this technique is different from what we heard from Malika yesterday and other people. Is because it's optical flow. It's really adapted to analyze dense labeling as opposed to single particle tracking. And so here are simulations that Itham and Roman did on artificial labeled particles here. So here you have a low density, and you can see that compared to the ground state, single particle tracking gives you less error, and so you have a more accurate estimate of the diffusion constants or other parameters. Now, if you increase the labeling here quite significantly, then you see that single particle tracking starts to be less concise than optical flow, just because it will not be able to identify all the densely labeled molecules here from one another. And so the estimate compared to the ground state of optical flow starts to be much better. And if you simulate something that is even closer to what we see with chromatin, meaning that you have densely labeled molecules, but then you have kind of clusters, clutches, hubs, whatever you want to name it, of even denser chromatin. And if you simulate this and analyze optical flow and single particle tracking, you see that you clearly have a superiority of optical flow, which gives you a much precise estimate of the different parameters. So this is what they used to analyze the vectors of movement of the intensity of all these thousands of pixels in each nucleus over hundreds of frames. And doing this, you can obtain trajectories for each pixel here. So it is color-coded for the number of steps. And you see that all these pixels have an orientation over time. You can define, of course, again, these domains, but you also have speed, orientation, and all the other parameters. So what is nice with this technique that we called high D for high dynamics is that you can map the global dynamics in the entire nucleus over thousands of pixels, depending on, of course, your resolution, and over several micrometers over time, over relatively long time intervals, which enables you to get true quantitative data here. For each pixel, you have a track. For each track, you can do mean squared deviations and can compare it under different conditions. And then you can analyze what you want. So here it was Bayesian that enabled us to define the diffusive behavior for each pixel, whether it's normals or anomalies. And then what you can do, if you have this information for each pixel, you can go back and plot it on top of the nucleus. So we obtain dynamic maps of the nucleus with parameters that are specific to each area. So it's spatially resolved. And now here are 5 different models that were used on anomalous and anomalous diffusion. And you can see that there's sort of mosaic-like distribution over the entire nucleus in this U2OS cell. From the diffusive behavior, you can extract parameters such as diffusion constant, anomalous exponent, or velocity. Let's just stick with diffusion constant. So you see that you can have diffusion constants that go from very, very, very small to super diffusive. And you can see these super diffusive patches here that are within the interior of the nucleus. And you have these dark blue, very slow moving, mostly in the rim. Now, to analyze this, having such a large heterogeneity is a little bit complicated. So what did is, what Item and Roman did is they used a general mixture model to define probabilities of being in different groups of motion. And so the model basically came up with that it's 3 areas that are most relevant for types of motion that we see. A low motion area, intermediate, and fast moving areas. So when we apply this to our U2OS nucleus, which is serum starved here, what you can see based on the maps of the diffusion coefficients, again, the general mixer model, you can see that you have the rim on the outside, this is red, very slow moving, and in the inside you have this mosaic of different areas that cover the entire nucleus. It's mainly anomalous and subdiffusive, but there's this mosaic that defines domains. So first question is, do these domains relate to chromatin density? In order to do this, you can measure the density of the labeling that you have in the nucleus. So it's something that was done by Kerstin Bystricky a number of years ago. So you can define the quantity of labeling in different areas. In purple, it is above a certain threshold, so let's call it heterochromatin or dense chromatin, and in green, it's below the threshold. So we traced the limit around these purple areas and plotted it on the dynamic map. And then ask whether within these purple areas of heterochromatin, it's exclusively intermediate or slow. And it turns out it wasn't. So whether you have euchromatin or heterochromatin, it depends on where you are, whether you're at the rim, so you're really slow. But when you're in the interior, it seems independent of chromatin compaction, whether you move in an intermediate or in a fast fashion. So these are U2OS cells. And when you look at other cell types, for example, our MCF7 cells here that I showed you before, you can again define the heterochromatin, euchromatin maps. You can find the diffusion coefficients. And when you overlay it, again, you see that these heterochromatin blobs do not necessarily all fall within the intermediate moving domains. They correlate at the periphery, but not within the nuclear interior. So dynamic domains, at least the speed with the extent of diffusion coefficient doesn't tend to correlate with chromatin compaction. Then we asked what happens when you activate transcription. So one way to activate transcription here is not in the specific genes, but is to add serum to serum-starved cells. So the cells are serum-starved. Again, they sit there, they're kind of arrested, synchronized. They don't really describe much. I mean, they're still alive, so there's a little bit of transcription, but not much. And then we add serum, so you expect many genes to start transcribing simultaneously. And just quickly show you this image here. We went through some trouble doing this. Just these two S cells, they do not really change the cell cycle. They start their cell cycle, so you have a little bit more here in G2. But what is, just to prove you that transcription is really starting, is the incorporation of pyronin. So you see here in dark, in black, the incorporation of pyronin in serum-starved cells, and in green in normally growing cells. And you see that when you add serum after a short period of time, you see the shift. So you really start making messenger RNA in these cells. And this is what happens to your dynamic map. On the left, no serum. Here with serum, you still have the rim that is dark red, so it is not moving. But in the interior, you can clearly distinguish a reduction of the yellow fast-moving domain. So the bulk of chromatin tends to move slower when you activate transcription. And that is totally in line with what we've seen on a single gene, on a single anchor tag cyclin D1 transgene. So that seems to be a global feature. And this is specific to chromatin, because when you look at RNA polymerase 2 here, so you have RNA polymerase 2 tagged. Again, it's dense labeling, just like chromatin, maybe a little bit less dense. You can define all the parameters I showed you before using high D. And then you look at the diffusion constants in the absence of serum, in the presence of serum. And when you block transcription, and you see what you expect to see, is that when you the polymerase moves less when you block X elongation. We also looked at the estrogen receptor itself. Again, you have dense labeling of this estrogen receptor, which is in these cells is GFP tagged. And we define diffusion coefficients. Here on the right-hand side, you have a quantification. In the absence of estradiol, the green line, you see a large variability of these diffusion constants. We add estradiol to it. You can establish new density maps, but the diffusion coefficient values themselves, they are much reduced when estradiol is added, when the estrogen receptor is bound to DNA. So you will see similar values as with the chromatin that is reduced in transcription. Now to finish, I would like to just put this in context. Why are we interested in estrogen receptor? Because so this is what this looks like, the ER-GFP in a cell using SIM microscopy, so high resolution, super resolution microscopy. When you add estradiol in the absence of hormone, so it's all diffuse here. When you add estradiol to it, you see these blobs appear, I mean, foci. So they kind of congregate. That's a little bit reminiscent of what I've shown you at the beginning in the progesterone receptor gene and its regulatory domain. The promoter, but you have many enhancers, and when you activate transcription, kind of come closer together. They create these sort of hubs that are around the gene to transcribe it. And this is also in the context of many years ago, we looked at different genes that are responsive to estradiol. And when you look at them, they're kind of all over the nucleus. When you add estradiol, they don't change their position. So the genes, they stay where they are, just like here in red, our cyclin D1 gene. It doesn't move, you know, you add estradiol, it's not gonna move around the entire nucleus to go somewhere to be transcribed, it stays. Because it's the estrogen receptor that starts forming clusters right next to it. Here, when you zoom in on this picture over time, the estrogen receptor is diffuse, and then you kind of clusters next to our label when in blue you make messenger RNA. So I believe that you have pre-organized structures, and then with time you cluster the transcription factors around it to help transcribe, but that will cage in the region and reduce the apparent motion. So in conclusion, I've shown you that chromatin motion is highly heterogeneous. Again, I'm talking about motion on like diffusive properties. We haven't looked at motion over times, over days, over hours. Here we look at motion over seconds and minutes. The subdiffusive, mainly subdiffusive domain dynamics are dependent on activity. Transcription occurs in pre-established domain organizations that do get reorganized and kind of caged in, and which reduces the amplitude of motion. And this is in human cell lines. With that, I like to thank again the people who were involved, my group. So here is Itham and Roman for the high-D techniques. 3D fish was done by Sylvia Kuchinova and Isabelle Goffon. The anchor tagging and single cell imaging by Thomas Jarnier and Ludmila Recoul. I also like to thank Alain Camgoué, a mathematician and informatician in the Institute, for the 3 loci survivor zone analysis, and our collaborators, Frank Lauder from NeoVertec, Mark Marty-Renum and Job Decker for the 5C analysis modeling, Tom Saxton for help with promoter capture high C, and Danielle for discussions and trying to help make sense of the reduced dynamics in the frame of contact prone movement. And our sponsors here, I like to thank, of course, the Nucleum Consortium for helping us publishing. And I just like to point out that we just moved in a brand new building. This is the Center for Integrative Biology in Toulouse, and check our website for upcoming postdoc group leader and other positions next year. And thank you for your attention.
Thank you very much, Kerstin, for these great talks. There are some questions. So, question from Srinivas Shulu Kurukuti. Have you seen any ER-induced interchromosomal interaction in live cells?
No, we haven't. We haven't looked in live cells. We haven't. No.
Okay, one question from Thomas Sabate. Hello. Did you try to use high-D into color imaging, for instance, with H2B to have chromatin dynamics, and with a transcription factor or RNA-2 in order to correlate directly in each cell the 2 dynamics?
Yeah, it's on the list. We'd love to do that, but of course we need because we're here the analysis at nanoscale. So you have to have a single image acquisition system, right? You can't do it sequentially, so it has to be simultaneously. So we are still setting this up. Or
eventually with H2B and MS2 tagged MS2 tagged cell line. But
yeah, that we are doing, but I haven't finished the analysis yet.
Okay, thanks. From Marcel Michali, in the experiment using cells released from quiescence, not only transcription is affected, but chromosomes also enter in DNA replication. How can you dissociate both effects on chromatin behavior?
Yeah, Marcel, that is some argument we had all the time, and this is why I showed the flow cytometry with the FACS analysis we did. So what we see is we do see an increase cells going into G2, but yeah, can't distinguish. It's a good question. In fact, we couldn't find any evidence for increased number of cells in S, but I mean, if some go into G2, it's likely that some from G1 also go. Yeah. It's very short. I mean, we add serum for 10-15 minutes only.
Okay, a question from Mathieu Lavigne. Mathieu?
Hello. Very nice work. I'm Matt Lavin. I'm in Greece in Fleming Institute. So I'm very interested in your experiment with the DRB. How long did you put DRB for?
10 minutes.
10 minutes. So yeah, you assume you still have some elongating, some RNA tethered to your chromatin there.
Yes, probably. Yeah, because
Okay.
you have the polymerase that kind of falls off. But yeah, the ones that are already engaged will be there.
Yeah, for long genes, you would assume you still have some RNA tethered. But have you tried to use amanitin instead, for example, where you know you won't have this problem of losing polymerases?
No, we used the other one. We used
Flavopiridol.
the drug that blocks initiation.
Triptolide.
Yeah.
Yeah.
Yeah. And
so
actually,
the
effects on the dynamics we see are not much different. Although in one case, you still have the elongating polymerase, in the other case, you still have the initiating one. Yeah.
Oh, that's interesting. So about the stiffness of your chromatin, are you aware of this bioarchive paper that came out from Mirnes and collaborators, where they sort of suggest that the nascent RNA coming out of the DNA is actually stiffening the chromatin?
Yes. Would love to test.
Okay, great.
Yeah.
So I think I'm going to contact you because I'm quite interested in those things you're doing. I think we can find some common interest. Thanks.
Talk to you soon. Thanks.
Bye.
Yeah, another question from Pavel Kos. Maybe I missed, but have you taken into account the cellular motion and its rotation when you measure MSD?
Have I taken into account the what?
The cell linear motion, yes, and its rotation when you measure MSD.
Yes, it's all corrected for drift, and everything is corrected for. There are like 20 some pages of supplementary material. Please have a look. Yes.
So another question from Vera Pancaldi.
Yes, hello. So this was very, very nice. Thanks for this great talk. So since we are at the end of the meeting, maybe we can get back to the questions we were having on the first day. So what do you think about the relationship between the 3D structure changes and transcriptional changes? In the light of the fact that some people say that in some states of some cells, there doesn't seem to be a link. Do you think it's because it's a different cellular state? Do you think it's because there are different scales?
I think it's different scales.
Okay.
To me, it's a difference in scale. That's why I insisted. So what we do is we really look at the physical parameters, like at very short time scales. So if we look at, and when you look at remodeling of TADs, to me, that is almost long range. But what we look at is within these topologically associating domains.
Right. But do you think it's about scales in space or also scales in time, I guess? Because what do you predict would happen in the long run? Your experiments are very short time intervals, right?
Yep.
So what do you think would happen in the end? Because I guess the cellular processes do take long time to, or at least they are established for longer times.
Well, I think we would need to find a system where in the longer run, let's say over a few hours, you actually see significant changes in expression profiles, right? Because as I said, these MCF7 cells, they are poised to activate transcription and they have no intent of changing anything. So if everything you look at will remain local. But if you look at the more of a cellular differentiation system, if there was something that we could look at within the microscopy feasible timeframes, I would expect the domains to also evolve.
Mm-hmm. Yeah. Okay, thanks.
Okay, 3 more questions. So from Ilya Flymer, have you tried quantifying ER clustering next to the expressed genes after addition of E2 in the experiment at the end of your talk?
Yes. Yeah, again, but it's not ready. These are very, very time and labor intensive experiments. Rick Young has done some work, and Ibrahim Sisse also on nuclear receptor clustering, and you can clearly see these phase separation type of behavior, and I think we see that also next to our gene, but it's very, very difficult to label everything at the same time.
Okay, so a question from Masiesh Kerin. So I don't know if I the pronunciation. So how do you reconcile your observation of decreased distance between phosphate probes following E2 treatment in MCF7 with results from Wendy Bickmore, where she sees increased distance between enhancer and promoter?
Yeah, I know. I've heard Wendy talk, and yeah, I don't reconcile it. Other than it's not the same gene. It's not the same locus, and yeah.
But it depended on the distance between the two. When they were far away, you have a colocalization, if I remember well.
We do see both. So what we see is some general folding over of the regulatory domain. But within the domain, you can have some enhancers. So we have different steps, actually. So you have some enhancers that come closer and then they come apart again. And at 45 minutes. And so between 45 minutes, some are closer. And then 3 hours, you can see them apart. And the folding over gets more intense. At 45 minutes, you have something that sort of happens. And at 3 hours, it's really kind of locked in. I think it depends on how long after addition of estradiol you look at it. It depends on the locus, although we have looked at other estrogen activated gene loci where we do see similar things as with the progesterone receptor. But yeah, what is nice is we have the progesterone receptor system, similar data both in 5C and the modeling and the fish data and super resolution. Everything kind of falls together. Whereas in most other loci, we only have one or the other of the experiments at really high resolution.
But so for your interpretation is in terms of chromatin fiber interaction. I don't know. It's in terms of interaction. Not on. You could imagine that the solvent becomes more viscous when you, or maybe it's crazy. When you express the genes, or maybe when you produce RNA, or when you have some protein, RNA protein, or you. So it. So your interpretation of the slowing down of the movement is in terms of interaction. That's right.
Now, I mean, we do also see a number of proteins that progressively pile up there. So you have estrogen receptor binds, but then what you can detect in TRPC at least becomes more. So you do have a quantitative accumulation of proteins. And we had this last week. There was a conference at the NIH who was dedicated to enhancer promoters, and it's clear that there are systems where you really increase proximity in enhancer promoters, and others where you actually don't need it. It's all mediated by whatever proteins you pile up in the middle. And I think with the estrogen receptor, we are somewhere in between. What I call caging in doesn't necessarily mean you do need direct contact.
Okay. Okay, so yeah, success. So one question from Daniel. Daniel.
Yes. I mean, but I can ask Kerstin later, otherwise. But I was curious about your transgene experiment where you measured the MSD. In your transgenes, do you have also the same complexity in terms of enhancers than the endogenous locus?
That's a very good question. I don't know. Because we haven't mapped really the area around it where we know where the transgene is located, but we haven't mapped the contact with other areas. Haven't done it. But the only thing I can say is that so the transgene is inserted via flip-in, the FRT system, so that we could vary the composition. So we also can insert a transgene that doesn't have the promoter, or doesn't have the gene, or doesn't have whatever. as a control. And we also have FRT tags at different places in the genome. And the effect on the mean standard deviation is the same no matter where you are in the genome.
Okay.
So I think the transgene is less sensitive to distant enhancers, but I don't know, maybe it hijacks enhancers wherever it is.
And regarding also these experiments, you do see at long times this decrease in dynamics, but at short times it seems that you have a slight increase. So do you think it's just I mean, how do you explain these differences? Is the difference in the short times, or it's?
Yeah, I don't know. We see this rupture at 8 seconds, 5 to 8 seconds. I mean, we have discussed this before, Daniel, and I'm a bit prudent in the interpretation since you do have all the PAR-B proteins there. So I don't want to draw hasty conclusions without having separated the sort of technical influence of the PAR-B protein there.
Yeah, okay.
So, yeah, but yeah, we should talk about it again.
Yes.
Okay, so since it's the last talk, we can continue a little bit if you don't mind the questions. We can finish the questions. So, question from Anna Compalance. Thanks for the very nice talk, Kerstin. It has been shown that the 8-oxo-G is induced at the promoter level, and that processing by BER pathway is important for the transcriptional activation in response to estradiol. So I can't understand, but have you tried to see what happens when you block the repair of the lesion?
No, you're talking about the paper by Gilles Salbert. No, I haven't looked at it. No. Okay. No, yeah. I actually studied it with the students the other day. I was also thinking we should revisit this. It has been a little bit neglected. You're right.
Okay, thanks. And I guess the last question from this conference from Geneviève Fourel. So could it be that nuclear actin fibers are involved in reduced mobility upon transcription?
Definitely. I have a feeling they are. Okay. And as soon as a new postdoc starts, we'll do that.